Hello YouTube! In this video I'm going to present an anti-realist interpretation of natural selection. So I'm assuming a general understanding of the realism-anti-realism debate. I have a lot of videos on this topic, if not. But realists take our best scientific theories to provide true descriptions of reality. Anti-realists see scientific theories as instruments for systematizing, predicting and controlling the observable phenomena, but not as uncovering truth beyond that. Now, before presenting this argument, I do feel the need to emphasise that I'm not claiming that modern evolutionary theory is in any way defective, nor does this argument provide any support to creationism or anything like that. I'll say a bit more about this in a pinned comment, because it would be tangential to go into it here, but I'm just flagging this up explicitly. I'm defending a philosophical position, not a scientific or pseudo-scientific one. The modern evolutionary synthesis is one of the most remarkable theoretical achievements in the history of science, and creationism is idiotic. So yes, in saying that this is an anti-realist or instrumentalist view, this is not in any way, that's not meant in any way as a criticism. Okay, so the argument begins with a problem concerning the relation of natural selection to biological individuality. The concept of biological individuality plays a very important role in selective models. There are three basic conditions required for natural selection to take place. First, the members of a population must exhibit variation in traits. There must be differences in morphology, physiology, or behaviour. Second, some of this variation must be heritable, so that with respect to these traits, offspring resemble their parents more than they resemble the population average. Third, there must be differential reproduction. Due to the struggle for existence, some organisms must be more likely to reproduce as a result of distinct traits. So, if trait T increases the probability of survival and reproduction, and if T is heritable, then selection will act over several generations to spread T throughout the population. Now, prima facie, all of these conditions invoke the concept of individuality, or the notion of biological individuals. In order to describe variation of traits, we need to distinguish different individuals in the population that bear those traits. Inheritance involves a trait from one individual being passed to another individual. Differential reproduction presupposes a distinction between reproduction and growth. So, both reproduction and growth involve the creation of new biological material, but reproduction is distinct in that it involves one individual giving rise to a new individual, whereas growth would be one individual changing over time. So, as a purely conceptual matter, natural selection, at least as it's standardly understood, requires populations of biological individuals. Furthermore, the concept of individuality has an important practical role in testing selective hypotheses, or like models of selection. Suppose we want to measure the fitness of a gene. Well, that may involve doing a genetic analysis on individuals of different generations, so we need to make sure that we sample each individual only once, and we need to assign each individual to the correct generation. Imagine if in measuring gene fitness in humans, you were to sample from a cell in my left arm and a cell in my right arm, right? Like if you sampled just human cells in a completely indiscriminate manner, or without imposing the right kind of conditions for sampling, you'd get the wrong answer concerning patterns of human evolution. You might sample 1,000 cells from one person and a single cell from another person. What we need to do is measure each human individual only once. So the basic point here is that biology often involves counting, and our concept of individuality tells us what to count. So it's unsurprising that one of the central questions in philosophy of biology is, what is a biological individual? I do have a couple of videos on biological individuality, so you can check those out if you want more detail. But it turns out there's a very wide variety of individuality concepts. Different people will define individuality in different ways. Also, I should say, just as a terminological point, sometimes biologists will use the terms individual and organism interchangeably. But in the context of natural selection, we probably want to avoid this because many entities that are not organisms can undergo natural selection. Viruses and prions are not usually classed as organisms, but they're certainly subject to natural selection. So when we talk about biological individuals here, we're using the term in the broad sense. Okay, with that said, here are just a few cases that challenge our intuitive understanding of individuality. So, consider a grove of quaking aspen. This initially appears to be a collection of many individual trees. So there are some aspen trees shown in this image here. We probably think of each tree as being a single biological individual. But these trees are all connected by a network of underground runners. So they're all physically connected in the root system. The grove expands as each tree sends out runners, which either grow into new trees or contact other runners and fuse with them. So all of the trees in the grove are physiologically connected by an underground root network, and they're all genetically very similar. And they're all developed from a single tree to which they have that genetic similarity. So biologists draw a distinction between the ramette and the genet. There are many ramettes, but a single genet, a single genetic individual, a single collection of organisms all genetically very similar. Obviously, we can't expect exact identity because mutations will accumulate over time, but there is genetic similarity. When a tree sends out a runner, is this growth of a single organism or a single individual, or is it a case of reproduction, a parent producing offspring? It's maybe not so obvious. It's interesting to compare the aspen grove to the oak tree. Each branch on an oak tree grows independently from other branches. So a mutation in a cell at the tip of the branch is passed on as the branch lengthens. But obviously it's all connected, in a single tree. Peter Godfrey Smith, in his book Darwinian Populations and Natural Selection, suggests that functionally oak branches are similar to aspen trees, and the oak tree is analogous to the whole aspen colony. So maybe the individual here is not the tree, but the whole collection of trees. Or consider eusocial insect colonies, such as honeybee colonies. These are often considered to be superorganisms. Honeybee colonies are tightly integrated entities containing tens of thousands of bees. Members of the colony have very sophisticated methods of communication, such as the waggle dance used for foraging, that helps maintain the functional integration of the whole colony. The colony exhibits a kind of physiological structure, a physiological integration, with insects altering the environment so as to maintain a stable temperature, stable gas concentrations within the colony. There's a strict division of labour, with bees organised into castes that perform different functions, and in particular the honeybee colony as a whole has a germ-soma distinction. Most members of the colony are sterile, because only the queen and the male drones reproduce, while female workers do not. So there is a reproductive division of labour, similar to how in the human body you have specialised reproductive cells. Like your liver cells are not going to reproduce, right? You're not going to have a child from your liver cells. You have specialised reproductive cells, the sperm and the egg. Similar in a honeybee colony, specialised reproductive parts. The colony also has something analogous to an immune system in that some workers act as guards. So basically, the whole colony is analogous to an organism, with the bees analogous to cells. Biofilms, such as dental plaque, are multi-species communities of bacteria that exhibit a number of interesting properties. Bacteria in a biofilm communicate through quorum sensing, and they also tend to share genes quite promiscuously. There are various mechanisms of lateral gene transfer, and in a biofilm there tends to be a lot of sharing of genes. The biofilm as a whole seems to have a life cycle. So it begins with attachment and colonization as the initial bacteria attach and then different bacterial species join, followed by growth, during which bacteria produce something called the extracellular polymeric substance, which structures the biofilms and contains enzymes to help the digestion of nutrients. And then finally dispersal, when the mature biofilm releases cells into the environment. After dispersal, the cells will spread, colonize new surfaces, and form new biofilms. So, the biofilm as a whole seems to have a life cycle, including reproduction. But is the biofilm an individual, a whole individual with bacterial cells as parts, or is it merely a community of individuals? So these kinds of cases are designed to challenge our intuitions about individuality. What is a biological individual? Is there any fact of the matter? And these are just a few cases, right? Like there are countless cases in the biosphere of entities that exhibit, very strange properties or at least they exhibit properties that challenge our intuitions about individuality. In an attempt to impose some order, biologists have proposed a variety of definitions of individuality. So here are just some of the proposals. I won't go through these in detail. Bottleneck of the life cycle. A new individual begins when the life cycle is reduced to a single cell or something else sufficiently small. Germ-soma distinction. An individual must have certain parts specialized for reproduction. Genetic homogeneity. Two cells are part of the same individual if they have the same genomes, or at least sufficiently similar genomes. Immunity criterion. The individual is demarcated by an immune system. The immune system distinguishes the parts that are self from the parts that are non-self. Sexual reproduction. An individual consists of all cells descended from a zygote. Okay, that's a small sample of proposals. Lidgard and Neihart, in their article The Work of Biological Individuality, list 24 definitions, many of which are very broad and would permit finer distinctions. So at this point, resolution of the debate in terms of a single definition is looking increasingly unlikely. Many definitions of individuality face pretty obvious limitations. For example, one obvious problem with most of the definitions I've just given is that they entail that there cannot be unicellular individuals. The immunity criterion is the only exception. The historical trend has been a proliferation of individuality definitions, the majority of which have remained in use. So of the 24 definitions in Lidgard and Neihart's article, some of them were proposed in the 1800s, all were still in use around the year 2000. In response to this, many philosophers and biologists have adopted a pluralist approach to individuality, where many different individuality concepts are legitimate depending on the organisms in question and the goals of the scientists studying them. The individuality concept provides a classification scheme, and different classifications will be appropriate for different contexts. The debate here mirrors the debate about the species concept. There's a huge variety of definitions of the term species, and these carve up the biosphere in radically different ways. This has driven some philosophers to suggest that many species concepts are legitimate. No single species concept is appropriate for all kinds of biological investigation. It's natural to suppose that if this kind of view works for the species problem, well, you can straightforwardly apply it to the individuality problem. There are many different legitimate individuality concepts appropriate for different kinds of biological investigation. But there is a problem here, which arises from the connection between individuality and natural selection. The problem is that if we adopt different individuality concepts, this will entail different answers about a variety of other matters. When measuring the fitness of a gene, different individuality concepts will lead biologists to count generations and offspring differently, leading to different judgments of the fitness of the gene. So if you take the rammet as the individual, the aspen rammet as the individual, well, allele A1 may be fitter than A2. But if we take the genet as the individual, A2 may be fitter than A1. Which is the fitter really? Well, if both individuality concepts are legitimate, then it looks like there's no fact of the matter here. Or consider arguments concerning group selection. Many biologists tend to reject group selection, often on the grounds that due to the far faster turnover of individuals relative to the groups of which they are a part, selection on individuals would overwhelm any selection acting on the group as a whole. Even biologists who defend group selection generally accept that it can occur only in limited conditions, like Maynard Smith's haystack model or Wilson's trait group model. But if many different individuality concepts are legitimate, there may be no clear way to distinguish a mere group from a genuine higher level individual. I mean, we want to say that clearly selection can act on humans, even though we are collections of cells. The issue here is because of these tight connections, because of the connection we saw between individuality and natural selection, there is a bit of a problem just embracing pluralism about individuality. Now, one obvious response here is to say, well, why not just allow that selection occurs on multiple types of individuals? We might say that the Aspen genet and its ramets are all individuals, and selection occurs both between different genets and within genets on the ramets. In fact, that's probably what we would want to say. That's probably what is actually going on. There are many different individuals, and selection can occur on all of them. I think this is basically correct, at least in spirit, though the solution of the problem is not quite so straightforward as this. The reason is, natural selection is a causal process. Now let's assume that many individuality criteria are legitimate. So there are many types of individuals, I1, I2, I3, etc. So a ramet is an I1 individual and a genet is an I2 individual, depending on what individuality concept we apply, right? Well, it seems then that to maintain realism about natural selection, we would need to show that selection on I1, I2, I3, and so on, for every individuality concept, delivers the right result about total evolutionary change when all of them are added together, when all are taken together. So in other words, we add the evolutionary change per the various individuality criteria to get the total evolutionary change. Just as in Newtonian mechanics, we take the sum of all the forces acting on an object to get the total net force on the object. So as Sober points out in his article Apportioning Causal Responsibility, it's always a sensible question to ask. For instance, what contribution does the gravitational force from a planet make to the acceleration of a particle? Right, we can precisely quantify the magnitude of the causal contribution of gravitational force. So in the case of Newtonian mechanics, we have a net force on a particular object, and then we can engage in causal decomposition. We can split that into component forces. But the causal decomposition of evolutionary forces is not possible given certain forms of pluralism about individuality. And this is because individuality criteria overlap. So the same entity might count as an individual per both I1 and I2. Or different individuality concepts might draw the boundary of the individual in slightly different ways. In Newtonian mechanics, it makes sense to ask, what is the relative contribution of the force of gravity and the force of friction to the net force on an object? We can exactly distinguish the different forces. But in a biological context, it makes no sense to ask, what is the relative contribution of selection on each of these individuals to the total evolutionary change, right? If you take the sort of force of selection per the different individuality criteria, it makes no sense to try to sum that to the total selective force. So consider humans, right? Humans are biological individuals, but there's some debate about what exactly counts as part of the human individual. The proper functioning of the human body is dependent on our close symbiotic relationship with many species of bacteria. And some biologists have suggested that the human individual is really the entire holobiont, consisting of the eukaryotic cells plus the symbiotic microbiome. Now, it would surely be absurd to try to decompose the total evolutionary change into the contribution of selection on humans per a genetic homogeneity criterion. So that a genetic homogeneity criterion would deny that the human microbiome is literally part of the human individual. So yeah, it would be absurd to try to decompose total evolutionary change into contribution of selection on humans minus the microbiome, plus the contribution of selection on the microbiome, plus the contribution of selection on the human holobiont, where the holobiont is, both the sort of normal human, as it were, plus the microbiome. This would be analogous to calculating the net force on a particle by adding the gravitational force of a planet and the gravitational force of that planet plus its moons. So, I mean, to view the same problem from a different perspective, I suppose we could ask, what exactly is the total evolutionary change? Well, as discussed earlier, what counts as evolutionary change is itself dependent on the individuality concept. Because, recall, to have evolutionary change rather than growth requires a distinction between reproduction and growth, and that's determined by the individuality concept. But different individuality concepts are legitimate in different theoretical contexts. So this is the problem. Individuality plays a central role in our models of natural selection, but there is no consensus on how individuality is to be defined. Indeed, the standard position is pluralism. There are a host of individuality concepts that are legitimate depending on our theoretical interests. Now, this prompts a simple response to the problem just outlined. In the context of studies of evolutionary processes, we might say that the individual is defined in terms of selection. In other words, selection is taken to be conceptually prior to the individuality criterion. On this view, then, there is a tight connection between selection processes and individuality, but the idea is that we identify the selection processes first, and then this determines how we classify entities into individuals. So selection comes first, right? And then once we've identified selection as occurring, this determines the classification of entities into individuals. Call this the selection first approach. And this is, I think, an appealing manoeuvre. Our concern here is natural selection in this theoretical context, in the context of developing selective models. It's arguable that a criterion of individuality that classifies individuals on the basis of the intrinsic properties of those entities can't be adequate, because whether or not an entity can undergo selection is dependent on its environment. A sequence of DNA that proliferates selfishly in one environment, such as a transposon in an organism's genome, may not do so in another. So just take the same sequence of DNA, but in a solution in a test tube or something, no selection would occur there. The DNA sequence is an individual in the first scenario, but it's not intrinsically an individual. In the test tube scenario, nothing would distinguish it from any other very complex molecule. So if our theoretical goal is to examine processes of natural selection, we can't classify individuals without specifying the wider environmental context. And it's then intuitive to say that the context that matters is just whether or not conditions are such that selection can occur. This kind of proposal has been defended by Ellen Clark in her 2013 article, The Multiple Realizability of Biological Individuals. Clark suggests that once we have identified where selection processes occur, we can then identify entities that exhibit individuating mechanisms. And I quote, "An individuating mechanism is a mechanism that either limits an object's capacity to undergo within object selection or increases its capacity to participate in a between-object selection process." So a certain class of entities counts as individuals where selection processes within each entity are suppressed, while selection between the entities is encouraged. So like in the human body, the human body contains various mechanisms for suppressing selection processes of our cells within the body. Obviously if cells start proliferating, selfishly, then, you get things like cancer, right? Whereas, there are, again, mechanisms that allow for selection between humans. Selection processes act on humans over generations. A similar approach is advocated by Peter Godfrey Smith he first defines the Darwinian population, which is a population that undergoes a selection process, and then a biological individual is just any member of a Darwinian population. Now, this selection first approach immediately dissolves the problem posed by individuality. Understanding of selection our understanding of natural selection isn't dependent on resolving the individuality question. Indeed, the theory of selection itself is what resolves the individuality question. Like once you have identified where selection is occurring, that tells you what the biological individuals are. So, to go back to the human example, like the reason why, say, cells in the human body, or at least some cells in the human body, are not biological individuals is because the within object, selection within the human body is suppressed by various mechanisms. So I think that something like the selection first approach is basically correct, but a bit more does need to be said about how exactly it's supposed to work. Recall that the initial problem was that it seems that identifying selection seems to require some criterion of individuality. For example, to say whether or not heritable variation obtains, we have to distinguish reproduction or replication from growth. Simply to insist that we identify selection first merely ignores the problem, doesn't solve it. The question is, how do we know when selection is occurring? We can grant that if we assume that some population is undergoing natural selection, then members of that population count as biological individuals. But how do we identify selection in the first place? Well, Clarke gives the example of aphid insects. During summer, females reproduce asexually, and at the end of summer, they reproduce sexually just once. Now, according to some definitions of individuality, particularly the sexual criterion, which holds that the individual is, and I quote, everything that develops by mitosis from a sexually fertilized zygote. So according to this definition, the entire collection of aphid clones is a single individual with scattered parts. And genuine reproduction only occurs with the sexual reproduction at the end of summer. This view of aphid individuality was defended by Jansen in the article What Are Dandelions and Aphids? Now, Clarke argues that when we examine the patterns of change within the collection of aphids, this shows that selection is occurring within the collection, right? And so we have to take the individual aphid insects as biological individuals because selection is occurring on them. She asks us to imagine that a mutation arises in the collection of aphid clones. The mutation improves the ability of each insect to survive and reproduce, and so it spreads through the whole clonal population. So suppose that the clonal population begins with 100% of insects carrying A1, but by the end of summer, the mutation is spread so that only 20% carry A1. To treat only the whole clonal population of insects as the individual would then give us the wrong answer about the heritability of A1 between clones. So since the whole clone began with 100% of insects carrying A1, we would expect this to be widely represented in later clonal populations that are produced sexually. But due to selection within the population on the mutant allele, most of the next clonal populations will carry that mutant allele, not A1. So in other words, if we ignore within clone selection, we will make the wrong predictions about later developments. I quote, within clone selection can cause informational decay by altering the similarity between ancestor and descendant clones. So, there is selection going on within the clone, and so we must treat the insects as individuals, not just the whole clonal population. We must treat each asexually produced insect as an individual, and so the sexual criterion of individuality is inadequate here. Now, I don't think this example is quite so persuasive. I mean, granted, there's no question that scientists must recognise that change occurs within the clone. There is, as Clarke says, informational decay, which will alter our predictions about the genetics of later clones. But it's not just obvious that this change must be treated as the result of an evolutionary process acting on generations of individuals. Why not, after all, treat it as change due to growth of a single individual? not all change is evolutionary change. Now, of course, in the aphids, we see, in this example, we see not just any change, but change in allele frequencies. So we might think, well, there's something special about genetic change which requires us to view it as evolutionary. But I think it's pretty clear that simply defining evolution in terms of genetic change is useful only in some contexts. So, for one thing, this definition entails, so to say, that just evolutionary change just is genetic change, entails that evolutionary processes could only occur in organisms that have similar mechanisms of inheritance as organisms on this planet. There's no reason to accept that, so a broader definition of evolution is required, especially in the context of astrobiology. Furthermore, we know that there can be non-genetic evolutionary change, even within life on Earth, through epigenetic inheritance mechanisms, for instance. For example, methyl groups can be added to the cytosine base of DNA, usually preventing transcription of DNA and so silencing the gene, and patterns of DNA methylation can be passed from parent to offspring, at least again in principle, right? This kind of thing could happen. So we shouldn't accept a genetic definition of evolution, at least not universally, and so we can't simply assume that any change in gene frequencies is due to evolution. Perhaps we should adopt a weaker kind of genetic view of evolution. We might say that genetic change is merely sufficient, though not necessary for evolution. After all, if the allele frequencies at some locus are changing, doesn't this show that the locus is not at Hardy-Weinberg equilibrium, which entails that some evolutionary force must be acting? But again, this, it seems to me, begs the question. The Hardy-Weinberg model tells us the conditions under which allele frequencies remain constant from one generation to the next. But the question of whether there are new generations within the aphid clone rather than just the growth of a single individual is precisely what is in question here. After all, in any multicellular organism, there will almost certainly be genetic changes over time just due to mutations as its cells divide. As the organism grows, it's in principle possible that very early in its life, 100% of its cells carry A1, and then by the end, 80% carry some mutant allele A2. So more needs to be said about what entitles us to assume that selection is occurring within the aphid clone, within the clonal population. Now, I do want to be clear here. My point is not to criticise the selection-first approach. I think that the selection-first approach is basically right. Rather, my point is, Clark seems to be assuming that as long as we can identify changing patterns of allele frequencies, well, that's enough to identify selective processes. And then, once we've identified selective processes, this fixes the classification of entities into individuals. My argument is that just as a conceptual matter, more is involved in identifying that selection is occurring. My point is just that any kind of change, including change in allele frequencies, could instead be modelled as growth rather than evolution. We can model it as changes in a single individual rather than generations of different individuals. Again, at least in principle. That's not what we would actually do, but the question is why that is, what justifies that. So my suggestion is that we can support the selection-first approach by drawing on a proposal by Robert Brandon in his article Adaptation and Evolutionary Theory. Brandon is concerned not with biological individuality, but with the role of the concept of fitness in evolutionary theory. The key explanatory idea of the theory of natural selection is that fitness, that is, well-adaptedness to the environment, is what makes an organism more likely to survive and reproduce. When we examine biological populations, we find a correlation between particular traits and reproductive success, leading those traits to spread through the population. For example, when we examine a population of birds, we might find that those with longer beaks tend to leave more offspring. Or we examine a population of moths, we find that those with darker wings tend to leave more offspring. What explains these correlations? Well, the answer is that certain traits make their bearers fitter than other organisms, or at least that's sometimes the answer. It is by making an organism fitter, by making it better adapted to its environment, that certain traits improve reproductive success. That's the key idea. And with this in mind, Brandon states a foundational law of evolutionary theory, which he labels D. So if A is fitter than B in environment E, then probably A will have more offspring than B in environment E. So the question then is, what exactly is fitness? What exactly is it for one organism to be better adapted to its environment than another? Well, Brandon argues that if D is to be acceptable as a universal scientific law, the definition of fitness needs to meet four criteria. First, fitness cannot be identified with reproductive success. This is because the theory presupposes that fitness causes reproductive success, or at least that fitness explains reproductive success. So we ask, why did A have more offspring than B? Because A was fitter than B. To identify fitness with reproductive success would make D a tautology. And furthermore, of course, we need to allow that reproductive success might be caused by processes other than selection, such as genetic drift. So the first constraint here is that we need to identify fitness with some property X that is independent from actual reproductive success. Second, D should be general, in the sense that it should be applicable to every population. This means that X must be a property that is found in all populations. So just a minute ago we imagined a population of birds where greater beak length improves reproductive success. Obviously we can't identify fitness with greater beak length because this definition would make D false for populations that don't have beaks, or populations where shorter beak lengths are favoured, or at least it would make D inapplicable, right, to populations that don't have beaks, inapplicable to any organisms without beaks. So we need X to be a property that's found in all populations. Third, D must be testable, in the sense that we must be able to tell how it is that D is to be applied to particular cases. One way to put this is that we must, in principle, be able to determine what it is for an organism to have X independently of its relative reproductive success, so that we can test D by checking whether organisms with X do in fact tend to have greater reproductive success. It may often be difficult or impossible in practice to determine whether an organism has X, but it's enough that there are some cases where this can be done. Although the identification of fitness with relative beak length fails the second criterion, it's obviously successful here because we know what it means for one organism to have a longer beak than another, and we know how to check beak length. Another one, might be like, if we think about properties like, strength and intelligence, and so on, and we know how to reliably identify these properties in organisms, at least in principle. Now compare this to the suggestion noted by Brandon that we treat fitness as defined to be close correlation with the environment. So we would say A is fitter than B in environment E if and only if A is more closely correlated to E than is B, or more closely correlated than B is to environment E. Well, how do we determine whether or not one organism is more closely correlated to the environment than another? This is not this is not an applicable definition. It's not one that makes B testable. So we need property X to be such that we can identify what organisms have X. Finally, D must be empirically correct. This is obvious. We must not give a definition of fitness that makes D false. It must be that the fitter organisms do in fact tend to have more offspring. If we were to identify fitness with relative intelligence, say, so that A is fitter than B in E if and only if A is more intelligent than B, well, yes, we might be able to come up with a way of measuring intelligence that is applicable to all species, so maybe it meets the first desiderata, but the first three desiderata, but it's obvious that there will be many situations where the more intelligent organisms will not tend to have more offspring. So those are the four criteria that we would expect for a universal scientific law. But Brandon argues that it is impossible to jointly satisfy these four criteria. And the reason is quite straightforward. Suppose we propose a definition of fitness that meets the first three constraints. So fitness is some property X that is distinct from reproductive success, that occurs in all populations, and that can, in principle, be identified in organisms. So we know what it is for an organism to bear property X, independently of its actual reproductive success. Now, what we can do is simply construct an environment, at least in principle we can construct an environment, that artificially selects against X. And since artificial selection is a form of natural selection, D is falsified. So just in case this point is unclear, I will state the same argument in a slightly different way. To avoid making D a tautology, we need to identify fitness with some property X that is distinct from actual reproductive success. Now, for D to be testable, which is the third criterion, it must be possible to determine, in general, whether one organism has X or whether one organism has X to a greater degree than another. But then we can specify a case where we artificially select against X by only allowing those organisms that do not have X to survive. After all, the point of the third criterion is that we be able to identify which organisms have X, and that allows us, in principle, to artificially select against X. So X does not promote reproductive success in our laboratory, under the conditions that we have imposed, and that means that we either reject the second criterion and hold that D is not general, that it doesn't apply to all populations, or we reject the fourth criterion and we say D is general but simply false. Well, we seem to have a bit of a problem here. One of the criteria must be abandoned. Brandon says we should give up the third. So he proposes the following definition of fitness. So he says A is fitter than B in environment E if and only if A is better able to survive and reproduce in E than is better able to survive and reproduce than B in E. This definition makes fitness distinct from actual reproductive success, because the fact that an organism is better able to survive and reproduce doesn't entail that it will actually have more offspring. I mean, by analogy, we can say that a car is able to go 150 miles an hour, even if we know that it never will actually go that fast. This is general in that it applies to every population, and it's not empirically incorrect. But what about the third criterion of applicability? Well, obviously there's not in general any way of telling whether one organism is better able to survive and reproduce than another. At least, distinct from our measurements of their actual reproductive success. So this definition of fitness renders D untestable. Under this interpretation, the reason why D remains important is that it can be used to generate specific hypotheses that are straightforwardly testable. D is a schematic law that must be instantiated by filling in the variables. So, for example, here's D again. If A is fitter than B in environment E, then probably A will have more offspring than B in E. We fill this in like so. If moth A is darker winged than moth B in polluted forest E, then probably moth A will have more offspring than moth B in that forest. So we instantiate D by specifying particular organisms drawn from a specific population for A and B. We specify a particular environment, and then we specify a particular trait for fitter. Now, what should you do if a specific hypothesis, like D1, is falsified? Well, obviously you don't reject D. Instead, you simply develop a different specific hypothesis, D2. So as Brandon says, and I quote, "Consider the instantiation of D concerning moths. If through experiments and observations it proved to be false, then our response would be, and should be, that we have incorrectly analysed the ecological situation." So we would create a different instantiation of D. D is a tool, an instrument, for generating specific models, specific hypotheses, which can be falsified. So it's a tool for generating falsifiable hypotheses, but it cannot itself be falsified. If we want to evaluate D, the question is not whether it's true or false, but whether it's useful for organising research, and whether it provides a useful unification of different specific hypotheses. I mean, the point of D is that without it, what we would have is just a whole host of unrelated descriptions of specific biological populations. D provides theoretical unity. So this is Brandon's view of the structure of the theory of natural selection. So how does all of this help with the individuality problem? What's the relation of this to our initial problem of individuality? So recall the issue, right? So natural selection seems to require a concept of individuality, but there are a whole host of different concepts of individuality. My plan is to defend a selection first approach. So the idea is that we identify selective processes first, and then this fixes the classification of entities into individuals. And with that in mind, I would suggest that the instrumentalist about natural selection would argue as follows. When we instantiate the general schematic law, D, with a specific hypothesis, such as D1, we must assume some particular classification of the biological world so that we have individuals that we can name for A and B. And all that is required here is that there are entities that we can count, where these entities can then be taken to produce other entities as offspring. And what is it that counts as offspring? Well, just any entity that's sufficiently similar to the parent entity, as it were, that's causally connected to the parent entity. So an aspen ramet can produce another aspen ramet, and so we can treat that latter aspen ramet as offspring as the first. We don't have to treat it as offspring as the first, we could treat it as growth instead, but the point is just where some entity X gives rise to another entity Y that is similar to X, it's possible to interpret X as having undergone reproduction. And that means that in principle this is a type of entity that can be used in an instance of D. Okay, then having stated an instance of D, so having formulated some specific hypothesis D1, the question is just whether this specific hypothesis is true, or at least whether it's sufficiently similar to the target system. And that's the question of whether it accommodates the data, whether it makes the right predictions. If it does not, then, as Brandon argues, we simply develop a different analysis of the ecological situation. And there are various things we might do, right? So, like, if to take the case of the moths, right? Like, we formulate this hypothesis that the moths with darker wings probably will have more offspring than the ones with lighter wings. But then, we go out there, we study the moths, and we find that actually the lighter winged ones are proliferating, or something like that, right? So in that kind of situation where our specific hypothesis has made the wrong predictions, there are various different things we can do to try to develop a new hypothesis. We might try specifying different traits as promoting survival and reproduction. Maybe it's lighter wings that are favoured, not darker ones. Or we might try changing the description of the relevant environment. Maybe there are relevant differences in different areas of the forest. But another thing you can try, at least in principle, is changing your interpretation of individuality, so that different types of entities must be used in the place of A and B. And this is what is going on in Clarke's aphid case, right? So Clarke's point is we can't treat the whole clonal collection as an individual because we then get the wrong answer about which collections will survive and reproduce, or at least we get the wrong answer about the fitness of genes over time as those collections survive and as those collections reproduce. So, in Clarke's aphid's case, her point is that there's a certain way of classifying the biological individuals in our selective models when we look at the aphids, which gives us the wrong answer, and we can fix that by adopting a different individuality criterion. So on this view, on the instrumentalist view, there is no need to adopt one definition of individuality. In most cases, biologists will presumably just adopt whatever individuality criterion is intuitive, given the species they're studying and their particular research interests, and then if what's intuitive doesn't work, well, try a different way of classifying the population, come up with a different specific hypothesis. Notice then that to insist upon a single criterion of biological individuality would tend to stifle biological research insofar as it would prevent biologists from exploring all of the possible selective models. Part of the incredible success of D lies in the fact that we can generate different individuality classifications. So, what exactly is a biological individual? Well, clearly this is a selection-first approach, insofar as we do not begin by specifying the individuals, rather we begin by developing selective models of a particular target population, and each of these models will invoke a particular classification of individuals in that population. The appropriate classification of individuals is simply a matter of which specific hypotheses work. Some specific hypotheses will make good predictions, others will not. There's nothing more to biological individuality than there is nothing more to a biological individual than its being a classification of a confirmed selective model. And this view is pluralistic. This might not be obvious. It might be thought that this kind of view is compatible with a monist account of individuality, because we might think, well, it's possible, at least in principle, that only one individuality concept will produce correct instances of D. So, you might think, well, like when we actually look at the correct instance of D, it might be that there is some single individuality concept that is being presupposed by them. There are two reasons to think that this is not going to be the case. So first, this ignores the role of different theoretical goals in the application of D. The appropriate individuality criterion will likely be dependent on, for example, the time scale and geographical range of interest. Consider again the aspen grove. If we are considering evolutionary processes over decades in a specific ecosystem, it would make sense to take the rammets as individuals, because the distribution of the rammets is likely to have significant ecological consequences in the short term. But if we're considering macroevolutionary processes, the genet view may be more appropriate, because asexual plant lineages may be less likely to adapt to the more substantial environmental disturbances which will occur over a longer term, and these lineages may also be more vulnerable to the accumulation of mutations that decrease fitness. I mean, I say may, this is, right, they may not, but if such hypotheses are correct, then the short-term success of a particular population of rammets is unlikely to have much impact on macroevolutionary patterns. In that context, in that specific context, we would probably favour an individuality criterion that picked out the genets as the individuals. So the appropriate individuality concept depends then on, what sort of, what range we're considering, right? What kind of range of timescales and geographical areas we're considering, and that of course is up to us. I mean, we have to decide where to draw the boundaries of our empirical studies. Second, a slight variation on Brandon's artificial selection argument shows that monism about individuality may be impossible in principle. Suppose that for some species, one particular individuality concept had been used to generate successful instances of D, where all other concepts had failed. Now, by making the right alterations to the environment, we could force a different concept. For example, with the aphids, we can generate successful instances of D concerning change within the whole collection of cloned insects, and this entails treating the insects as individuals. That's Clarke's point. However, we could structure the environment in just the right way so that hypotheses about the whole clone, that selective hypotheses about the whole clone are successful instead. One obvious way to do this would be to prevent any mutants that arise from breeding sexually. Then, even if the mutant allele spreads through the population via non-sexual reproduction, it will not be passed on to the next sexual generations. In the long run, the mutant allele would be a dead end. Indeed, the mutant insects here would be playing a role much more analogous to that of somatic cells in metazoans. Unlike in Clarke's original example, in this case, the sexual criterion of individuality would not give us the wrong answer about the clone-level heritability of the original allele A1. So, we have a pluralism about individuality. There are no general constraints on which individuality concepts can be used in selective models, and we should not expect a single individuality concept to be substantially more successful than any other. This is a deflationary pluralism, because it turns out that individuality is not a substantive property, but simply a matter of whatever classification scheme works in applications of the schematic law D. Biological individuals are not natural kinds. The biological individual is a feature of the selective model, not a feature of the world. Biological individuals are instruments, in the same way that D is an instrument. But notice that we are not committed to anti-realism about the specific selective models, the specific selective hypotheses. So the key idea here is that an instrumentalist interpretation of D and an instrumentalist interpretation of individuality are compatible with realism about specific hypotheses D1, D2, etc., in the sense that we can take at least some of these specific hypotheses to be true. And this is because these specific hypotheses, we can judge them as being more or less, accurately descriptive of populations in the world. So consider, for example, if aspen rammet A has darker leaves than aspen rammet B in environment E, then it will probably have more offspring in that environment. Well, this hypothesis may be true, and it may accurately describe what's going on in the world. It doesn't matter whether the aspen rammet is a genuine individual. Indeed, it doesn't matter what a genuine individual even is. Individuals are defined in the model. They are theoretical kinds. The models are applicable to the world just in case we have entities that we can count that correspond to these theoretical kinds. And it's clearly the case that we can count aspen rammets, and that these aspen rammets produce other aspen rammets that can be counted as offspring. And these entities are still real, right? Aspen rammet A and aspen rammet B really exist mind independently, and it is really true that A is more likely to survive and reproduce due to its darker leaves. So there's still a realist interpretation available for the specific hypotheses. And this is good news for the instrumentalist. As I noted at the beginning, the point of anti-realism, at least the brand of anti-realism that I favour, is that scientific theories are useful instruments for systematising, predicting and controlling the observable phenomena. So although theories are instruments, they do need to make true claims about the observable world. After all, if they failed to do that, they wouldn't be very useful instruments. So what's been presented here is an anti-realist instrumentalist interpretation of the theoretical framework, the underlying law of natural selection in D and the theoretical objects, namely the biological individuals, but an instrumentalist interpretation of D and of individuality that's compatible with making true claims about the observable world. We observe aspen rammets and aphids and moths, and we can make true claims about them. Okay, so the suggestion is that the model of evolutionary theory developed by Robert Brandon is useful for thinking about the problem of individuality. One benefit here is that Brandon developed his model for other purposes. He was not concerned about the individuality problem. So the application of his model to the individuality problem provides a rather pleasing theoretical unification. In this section, I want to provide some further support for this approach to natural selection by showing how it resolves three further problems relating to the relation between selection and individuality. First, origin of life and major transitions. So it was argued at the beginning that natural selection requires a concept of individuality because the concepts of heredity, reproduction and variation within a population all appear to depend on a concept of individuality. Now, a major problem which arises here, pointed out by Akasha in his book Evolution and the Levels of Selection, is that whatever definition of individuality we adopt, it seems that individuality will itself turn out to be a product of evolution. Early biological entities presumably would have exhibited less physiological cohesion, they would not have exhibited a germ-soma distinction, they would not have had an immune system, etc. All of these traits must have evolved, and natural selection must have played a significant role here. But then if natural selection acts on individuals, and if individuality is defined in terms of one of these evolved properties, it will be impossible to explain the emergence of this property, or at least it will be impossible to explain it in terms of natural selection, which is probably what we would want to do. So any substantive definition of individuality runs into some pretty serious difficulties in the context of studies of the origin and the early major transitions of life, the emergence of major properties like germ-soma distinction and immune system and so on. Well, the instrumentalist view developed in the previous part provides an easy solution to this problem. Even the simplest biological entities can still be countered, and they still produce other biological entities similar enough to them that can also be countered, that can be treated as offspring. That's enough to generate successful instances of D. There is no point at which individuality evolved. There are many kinds of individuality corresponding to the many kinds of properties that biologists might use to create a classification scheme. Most of those properties will be products of evolution by natural selection on earlier entities, but we can understand selection on those earlier entities by applying a different individuality concept, a concept that is not defined in terms of the evolved properties. Second, biological practice. It has been noted that while the concept of individuality seems to be central to many biological theories, the problem of individuality is of little concern to most practicing biologists. See Kovarka's article, Biological Individuality and Scientific Practice. The question then is, why is this? And are biologists justified in ignoring the problem? There is a minor mystery here, given the role of individuality, the central role of individuality in biological theories. Well, Kovarka seems to adopt a kind of selection-first approach to explaining this. She says of Clark's aphid case that biologists do not need to worry about the individuality problem because, and I quote, "As soon as a biologist realizes that evolutionary change can occur within a swarm of clones, she can infer that counting swarms rather than single aphids is a bad method of investigation." However, as I argued in response to Clark, the question is what exactly justifies the assumption that evolutionary change is occurring here. So yes, right, if you assume that evolutionary change can occur within a swarm of clones, then you're going to want to count single aphids. But the question is what justifies that assumption. Well, on Brandon's model, on the instrumentalist view, all we need to do is generate successful instances of D. Instances of D that accommodate the data and make good predictions. That's clearly possible. It may be the case that if one aphid insect is darker than another, it will probably have more offspring, or in other words, it will probably produce entities similar to itself. There is evolutionary change because the central schematic law of evolutionary theory can be successfully applied. That's it. Biologists do not think carefully about the concept of individuality because biological individuality is not a substantive property. There are numerous acceptable individuality concepts, numerous acceptable ways of classifying the entities in a given ecosystem. The right classification follows from the construction of a successful selective model. So, this is what biologists will concern themselves with doing, right? Biologists should concern themselves with constructing empirically successful selective models. Which is what they actually do. So basically the question is not what is a biological individual, but rather for any given biological system, right, what are the processes of selection occurring in that system? And then once you've developed an empirically successful model, that fixes the classification of individuals. But naturally you're not going to find biologists being too concerned about individuality concepts. Their concern is going to be developing testable and hopefully empirically successful selective models. Third, the problem of the paradigm. A difficulty for standard theories of individuality is what Matt Harber, in his article Colonies Are Individuals, calls the problem of the paradigm. This consists in the assumption that certain entities, usually medium to large metazoans, are paradigmatic individuals, and then in viewing all other entities in terms of their similarities and differences to these paradigmatic individuals. Harber suggests, for example, that treating eusocial colonies such as honeybee colonies as superorganisms often rests on the assumption that the organism is the paradigm individual, and that other entities are individuals only if they are sufficiently similar to organisms. So discussions of honeybee colonies tend to emphasize properties like the germ-soma distinction, which are found in paradigm organisms, organisms like humans, dogs, elephants. So the assumption is like humans, dogs, elephants, monkeys, etc. Those are the paradigm organisms, right? And then we, having selected these paradigm organisms, we identify what makes these organisms individuals. Maybe we want to say a germ-soma distinction. And then we look at the sort of vaguer cases like honeybee colonies and see if they also have that property. Now, the problem with this approach, Haber raises two objections to this way of approaching individuality. He says, first of all, this is just going to obscure the many differences between colonies and organisms. By sort of kind of classifying them in terms of this single shared property, right, that we've drawn from the paradigmatic individuals, we're obscuring relevant differences. Second, and more importantly, Haber says that this kind of approach is incompatible with, or at least in tension with, the population thinking that characterizes modern biology. A key feature of modern biology is the rejection of the idea that biological variation should be seen as a deviation from a natural state or a natural type. So the attitude that we have today is variation is ubiquitous, and there are no underlying types or essences that impose order on the biological world. So check out my video, Natural Kinds Essentialism, for more on this. But the basic idea is that whereas in the past, the biological world tended to be organised in accordance with certain essences, you would view a species as having a certain essence, and then particular organisms in that species as deviating more or less to a more or less degree from that essence. Today, we just have ubiquitous variation, and there are no underlying types or essences. So there are no paradigm individuals. Individuality can emerge in multiple kinds of ways. And so most of the substantive definitions of individuality will face this problem insofar as they're going to specify certain properties as being central to individuality. By doing that, we obscure the fact that there's just indefinite variation in individuality. In addition to the points Haber raises here, the sort of standard approach to individuality, the view that individuality is a substantive property, does face, I think, a problem of arbitrariness. Because even if we think that there are like paradigm individuals, the question is, what's the justification for treating a particular type of entity as the paradigm individual? So usually it's medium to large metazoans that are treated as paradigm individuals because those are the individuals with which we are most familiar, right? As we noted earlier, many of the individuality criteria that have been developed with metazoans in mind are inapplicable to unicellular organisms. If entities like bacteria were taken as paradigm individuals, well, we might identify very different properties as being central to individuality. But then why not, right? Why not take them as being the paradigm individuals? Microbes have been around for far longer and by various measures are far more evolutionarily successful than are, large animals. So, why not take them as the paradigm individuals? There's at least an issue of arbitrariness here, it seems. And if we think of individuality as a substantive property, it is difficult, I think, to see how the problem of the paradigm can be avoided. But the problem of the paradigm does not affect the instrumentalist view. On the instrumentalist view, we need not suppose that any particular property is necessary for individuality, or that any entities are paradigm individuals, whereas other entities are, lesser individuals that deviate from the essence or type of individuality to a greater or lesser degree. There are no paradigm individuals. There are just various kinds of individuals. We adopt a particular classification scheme for the purposes of developing selective models. Individuals, biological individuals, are those entities that appear in successful instantiations of the schematic law D. And then it's a straightforward empirical question, exactly what properties allow the entities to play this role. So the instrumentalist approach provides a unified treatment of all individuals, but in a way that respects the insights of population thinking. So, to sum up, The concept of individuality and the concept of selection are intimately connected. Selection acts on populations of individuals. Applying the model of evolutionary theory proposed by Robert Brandon, we can see that in the context of evolutionary studies, there is no need to have a definition or set of definitions of biological individuality. An individuality concept provides a certain way of classifying the biological world. The biological individual can be, I suppose, thought of as something that plays a functional role in that law. A classification of entities into individuals is acceptable if this classification is presupposed in empirically successful instances of D. There are good reasons to expect that different instances of D will presuppose different classification schemes, so we have a robust pluralism about individuality. Thus, from the war of ideas, from the conjecture and refutation of specific models, the central concept in the theory of natural selection, namely the construction of biological individuality, directly follows.